So whenever a cell wants to divide, it needs to round up in a process called mitotic cell rounding. This happens in our bodies, but also in tissue culture, in cell culture. And the process called mitotic cell rounding, the cell rounds, the DNA is being condensed, and eventually the DNA segregates into the two nascent daughter cells. So it's known that the cell rounds up, and it knows, and it's known that below a certain height of confinement, this rounding does not happen anymore. But until recently, we were not able to quantify the forces involved in this process. So what we do now in this experiment, using a confocal hooked up with an atomic force microscope, similar to the one we see here, we confine cells about to undergo mitosis with increasing forces. And we find that the higher this force of confinement is, the less likely the cells are to undergo mitotic cell rounding or to then undergo cell division. And we find that at a certain confinement force of about 200 nanonewton, the cell will not be able anymore to divide, and it would just sit there endlessly. And we can see that the chromosomes, which usually are being pulled towards the metaphase plate, are spread out all over the cell body and cannot be segregated nicely. We can measure the forces involved in this process, and at the same time we can image GFP labeled proteins or cherry labeled proteins in our cases. So when we confine a cell and it starts to undergo mitotic cell rounding, we can see how the tubulins labeled in green here try to collect all the chromosomes which are labeled in red here. Usually this process takes 30 to maybe 45 minutes, but in this confined cell this takes much longer, since the chromosomes are much slower in being condensed into the metaphase plate before they're actually then being segregated into the two nascent daughter cells. When you confine the cell more with higher forces, this process will take even longer, and eventually the cell will not be able to collect all chromosomes, and mitotic cell rounding is stalled and the cell will not divide. Okay, so to do our experiments, we use an inverted light microscope hooked up to a confocal unit, which allows us to look at the cell as it undergoes mitosis and to image the fluorescent probes that our proteins are labeled with. At the same time, we have an atomic force microscope, which helps us to exert forces on the cell and at the same time measure the mechanical feedback that the cell exerts on this confining cantilever. So the cantilever, which allows us to confine the cell, is kind of neglecting the 10-degree angle that we usually have with commercial cantilevers by allowing us to confine the cell between two parallel plates. So what we do is we use a normal thick cantilever, cut away whatever we don't need, and we're left with a thin cantilever on which at the end there's a wedge-like structure. And when we tilt this by 10 degrees again, the little wedge-like structure will allow us to have parallel plate confinement of the mitotic cell.